On March the 16th, 2020, a deep-sea robot descended through black water off Western Australia and found what looked like a glowing rope arranged in a vast spiral. The outer coil alone was estimated at about 47 meters. One later calculation put the full length as high as 119 meters, longer than a blue whale, possibly longer than any free-living animal ever recorded, but the camera had not found a sea snake, a worm, or a single giant body. It had found thousands of animals permanently joined together, each one doing only part of the work of staying alive. So what exactly was moving through the darkness, one animal or an entire society? The answer forces us to rethink where one creature ends and another begins. The machine staring at it was called Submersible, a remotely operated vehicle built to explore water far beyond the reach of divers. Above it floated the research vessel Falkor. Below it lay the submarine canyons of Ningaloo, where the continental shelf falls away into a cold, lightless world. The expedition was led by scientists from the Western Australian Museum, with researchers from several Australian and American institutions. They were there to survey a region that had barely been seen. During 20 dives, Submersible explored for more than 180 hours, sometimes descending thousands of meters. The team encountered glass sponges, octopus squid, sea cucumbers, and animals that may have been completely new to science. Then the lights caught the spiral. It was so large that the pilots could not frame the whole thing in a single view. A thin, translucent line curled inward like a galaxy drawn with living glass. Delicate tentacles hung beneath it. The colony was identified as an Apolemia siphonophore, a relative of jellyfish, corals, and sea anemones. The expedition announcement called it an estimated 150-foot animal and described it as seemingly the longest ever recorded. That wording matters. Seemingly. Measuring a transparent, moving animal in deep water is nothing like stretching a tape measure beside a whale. The spiral was drifting. Its body could contract and extend. Parts overlapped in the camera image. The most conservative, widely reported estimate described the outer ring at roughly 47 meters. Guinness later listed a reconstruction of the entire coil at up to 119 meters, but even that record notes that the full colony could not be directly measured. There is another competitor. In 1864, a bootlace worm reportedly washed ashore near St. Andrews, Scotland, and measured more than 55 meters. The record still appears in reference books, although ribbon worms are extremely elastic, and the old specimen may have been stretched beyond its normal living length. A lion's mane jellyfish has also produced tentacles longer than a blue whale, depending on whether you count a colony, an elastic body, or the farthest tip of a tentacle. The winner changes. That sounds like a technicality until you look closely at the Ningaloo animal. Then the argument becomes the entire story. A siphonophore does not begin when unrelated creatures meet and decide to cooperate. It begins the way you did, as one fertilized egg. That egg becomes a larva. The larva develops a stem in specialized growth zones. From those zones, new bodies bud outward as genetically identical copies called zooids. They remain physically connected. They share fluids, signals, and the consequences of every successful hunt. Yet each zooid is multicellular and corresponds in evolutionary terms to the kind of body that could once have been an independent animal. The colony grows by making more animals, then turning those animals into body parts. This is the first mental shift. A siphonophore is not a crowd holding hands. It is closer to a body whose organs still reveal that they were once individuals. At the front of many species sits a gas-filled float called a pneumatophore. Behind it may be a row of swimming bells called nectophores. Each bell resembles a tiny jellyfish and fires a jet of water. Farther down the stem hang feeding zooids, digestive zooids, protective bracts, reproductive zooids, and branching tentacles armed with stinging capsules. One part moves but cannot eat. Another catches food but cannot swim. Another produces eggs or sperm but cannot hunt. Separate one from the colony and it usually cannot live for long. Together they behave with the unity of a single animal. The familiar Portuguese man-o-war is built on the same basic principle. Its blue float catches the wind while tentacles trail beneath the surface, sometimes for tens of meters. What beachgoers call a jellyfish is already a floating partnership of specialized clones. The giant deep-sea species take that plan and stretch it into something almost impossible to see as a whole. For centuries, that construction confused even the people trying to classify it. Carl Linnaeus formally described the Portuguese man-o-war in 1758, when naturalists still grouped unfamiliar marine creatures by broad similarities in shape. Surface species could be collected from ships or found stranded on beaches, but the long, delicate forms from deeper water rarely arrived intact. During the 19th century, ocean voyages brought back more pieces. Naturalists described floats, bells, and feeding structures that looked as though they belonged to different animals. Some interpreted the colony as a chain of jellyfish. Others saw modified polyps. In 1829, the German naturalist Johann Friedrich von Eschscholtz established the order Siphonophora, giving these confusing drifters a scientific home without fully settling what the whole thing represented. The name comes from Greek roots referring to a tube and a bearer, a reference to the structures early researchers could see. Yet names based on fragments encouraged a fragmented view. A species might be reconstructed from specimens damaged by nets, preserved in jars, or collected at different stages of life. Even today, siphonophore classification depends heavily on the exact sequence and shape of zooids along a colony. This history explains why the Ningaloo footage mattered beyond its size. Scientists were watching an intact giant in its natural posture. The spiral, the spacing of its units, and the curtain of tentacles were not guesses assembled on a laboratory table. For a few minutes, the colony could be studied as the moving animal that evolution had built. And length is only the least strange result. Imagine trying to row a boat with a dozen people who cannot see one another. If every rower pulls with equal force at the same time, the boat moves forward, but it cannot turn with precision. A siphonophore solves this problem without a brain. Research on the species Nanomia bejuga found that its swimming bells divide the labor by age and position. Older, larger bells contribute more forward thrust. Younger bells placed farther from the colony's centerline provide more turning force. Their smaller jets have greater leverage, like tugboats pushing at the edge of a ship. By activating different bells, the colony can accelerate, rotate, reverse, or correct its course. The pattern is not improvised after the colony forms. It is built into the way the zooids butt along the stem. Development creates the engine and the steering system at the same time. Signals travel through nerve networks and large nerve fibers running along the shared stem. Each swimming bell has its own sensory and motor structures, but it also exchanges information with the colony. There is no commander sitting in a central head. Coordination emerges from a network spread across many connected bodies. That detail changes the picture completely. The giant spiral at Ningaloo was not a passive string being carried by the current. It was a distributed machine, able to coordinate thousands of repeated units while occupying a body plan longer than the largest whale. But propulsion is only one half of its design. The other half hangs below the stem like an invisible curtain. Large deep-water siphonophores often live between roughly 300 and 1,000 meters, where turbulence is weaker and sunlight fades into permanent dusk. In that calm water, a long colony can spread an enormous net of tentacles. Tiny side branches called tentilla carry batteries of nematocysts, microscopic capsules containing coiled barbed threads. When a copod, shrimp, or small fish touches the wrong filament, the capsules fire in a fraction of a second. Barbs penetrate or wrap around the prey while toxins immobilize it. The tentacle contracts and delivers the catch to a feeding zooid with a mouth. Digestion begins there, but the energy can move through the connected gastrovascular system and support zooids that never eat for themselves. One mouth feeds a body that may be many meters away, or depending on how you count, one animal feeds thousands of bodies. The tentilla are not generic weapons. Across siphonophore species, their size, shape, and arrangement vary with diet. Some are suited to soft gelatinous prey, Others form dense batteries capable of gripping hard-bodied crustaceans. The colony can hang nearly motionless, allowing tiny currents and the movement of prey to bring food into the net. In the deep sea, where calories are scattered through an immense volume of water, length becomes feeding area. That may help explain the giant spiral. By curling through a wide horizontal space, the colony can place thousands of tentacles across water that prey must cross. It resembles a drift net, except every strand can sense contact, fire venom, contract, and route the catch toward a mouth. The shape is both body and hunting behavior. There is a limit to the comparison. A human fishing net is useful only because someone watches it, repairs it, and hauls it in. The siphonophores net is alive at every point. Damage may cost it zooids, but the modular colony can continue functioning if enough of the integrated system remains. New units keep budding as old ones age. Its prey rarely sees the full danger. Natural light at several hundred meters is faint and blue. Transparent tissue erases much of the colony's outline. A small crustacean may encounter only one thread, one lure, or one flash. The enormous hunter does not need to chase in the way a shark does. It turns the surrounding water into part of the trap. Some siphonophores take the trap even further. Scientists studying the deep-sea genus Arena found tentilla tipped with red glowing structures. In video, those lures twitch in a way that resembles small swimming prey. Red bioluminescence is rare in the deep sea, and many animals there are less sensitive to red wavelengths. The lure appears to exploit that darkness, attracting fish toward a nearly invisible curtain of stinging cells. The colony becomes a fishing line that grows its own hooks, produces its own bait, and shares every catch across a living supply network. That sounds like an organism, yet every hook, mouth, propeller, shield, and reproductive unit is also a zooid. The categories keep collapsing. Pay attention to what happens as the colony grows, because this is where the idea of an individual becomes hardest to defend. In many siphonophores, new zooids arise in precise, species-specific sequences. A small bud subdivides into the members of a repeating unit. One branch becomes a feeding polyp, another develops reproductive structures, others form protective or digestive parts. The colony does not simply add identical beads to a string, it repeatedly manufactures a miniature team arranged in the correct order. Near a growth zone, the youngest zooids are packed together. Farther back, older units spread out and mature. The body therefore carries a visible timeline of its own development. New parts are forming at one end while older reproductive units may be released from the other. Some species even detach complete clusters called eudoxids. These free-living units carry feeding, protective, and reproductive zooids and continue a separate phase of the life cycle. A colony can therefore produce something that looks briefly like another individual, not by giving birth to a miniature copy of itself, but by letting go of an organized piece of its own body. There is a strange symmetry here. Humans build complexity by arranging specialized cells into tissues and organs. Siphonophores build complexity by arranging specialized animal bodies into something that functions like tissues and organs. Evolution reached toward the same problem from a different level. If you are enjoying this descent into the deep ocean, subscribe. We follow the animals that make ordinary biology feel unfinished. The comparison with a human body is useful, but it can also mislead us. Your heart and liver share the same DNA because they grew from one embryo. They perform different jobs, depend on one another, and cannot survive alone. By that logic, siphonophore zooids sound like organs, but zooids are not merely lumps of specialized tissue. A feeding zooid is organized as a polyp with a mouth and tentacle. A swimming zooid is organized like a medusa, the bell-shaped form associated with jellyfish. Each is homologous to an entire solitary animal body and related cnidarians. Evolution did not invent a new organ from scratch. It modified complete ancestral body plans and locked them into permanent dependence. So are zooids individuals? Biologists use several answers at once. Genetically, the colony is one clone descended from one fertilized egg. Developmentally, it is one continuous product of growth. Physiologically, it shares transport and coordination. Ecologically, it hunts and moves as one unit. But morphologically and evolutionarily, it contains many animal bodies. The disagreement is not a failure of science. It reveals that the word individual was designed for familiar creatures with one mouth, one brain, and one obvious skin boundary. Nature never agreed to obey that definition. An aspen grove can spread underground as one genetic organism while producing thousands of trunks. A coral reef is built by connected polyps. Social insects can behave like a superorganism, although each ant or bee can walk away. Slime molds spend part of their lives as separate cells and later assemble into a coordinated body. Siphonophores occupy an especially unsettling point on this spectrum because their zooids cannot simply leave. Their individuality has been traded for specialization. This is where language starts to smuggle in assumptions. Colony usually suggests members that existed first and joined later, like birds in a nesting colony. But siphonophore zooids never live through that independent stage. They bud in place from shared tissue. Organ usually suggests a structure that evolved below the level of a complete animal body, but a nectophore still carries the architecture of a medusa, while a gastrozooid carries the architecture of a polyp. The colony is therefore not halfway between a crowd and a creature in any simple sense. It is a different route to creaturehood. Natural selection can act on the success of the entire connected clone. A swimming bell that sacrifices feeding ability can still pass on its genes because the reproductive zooids carry the same genome. A feeding zooid can abandon locomotion because other zooids move it toward prey. Once every member is genetically aligned and physically trapped in the partnership, extreme division of labor becomes possible. That alignment removes one of the biggest problems faced by ordinary societies. Ants in a colony are closely related, but they still have separate bodies and at least some potential for conflict. Siphonophore zooids are clonal components that share circulation and fate. If the stem dies, no swimming bell escapes to start again. Cooperation is no longer a choice or even a behavior. It is anatomy. That trade made extreme length possible. A whale must keep every cell supplied within one enormous body. It needs thick muscle, blood vessels, organs, bones, and enough energy to move that mass. As the body grows, support becomes expensive. A giant siphonophore follows a cheaper blueprint. It is mostly water, extremely thin and modular. It can add another repeated set of zooids by budding along a stem. Length does not require scaling every organ into a heavier version. It requires adding more modules. The result can be 40 meters of animal with a body only about as thick as a broomstick in some giant species. It wins the length contest by refusing to play the whale's game. Of course, that design carries a cost. Siphonophores are so fragile that nets often tear them into pieces. For much of scientific history, researchers hauled fragments to the surface and tried to reconstruct the whole creature from disconnected parts. A swimming bell might be described separately from the feeding section it once propelled. Pressure changes and rough water could destroy the original arrangement before anyone saw it intact. That is why remotely operated vehicles changed the field. A robot can hold position beside a colony, film it in place, and collect delicate samples with far less damage than a trawl. The deep sea stopped arriving only as wreckage. Scientists could finally watch the full system move. Even robots distort the scene. Their bright lamps illuminate animals adapted to near darkness. Thrusters stir water around bodies that normally drift in calm layers. A manipulator arm can tear a colony with one careless movement. Researchers must choose between observing from a distance and collecting the tissue needed for genetics, anatomy, and formal species descriptions. This creates a strange scientific imbalance. We may have hours of video showing a spectacular colony that cannot be named a species with certainty because no diagnostic specimen was recovered. Or we may have preserved fragments with excellent microscopic detail but no record of the complete living shape. The animal exists across two kinds of evidence that rarely arrive together. New imaging methods are beginning to narrow that gap. High-resolution cameras reveal the rhythm of individual swimming bells. Laser references and stereo imagery can estimate dimensions in open water. Gentle collection systems can isolate part of a colony without crushing every soft structure. Genetic sequencing can connect a fragment to relatives whose complete forms are better known, but the basic challenge remains. A creature that is mostly transparent, easily torn, and distributed over tens of meters does not fit neatly into a specimen jar. Its biology resists the methods that built much of zoology, and the full system does not always look like a string. Some siphonophores coil into spirals to deploy their tentacles through a wide volume of water. Others contract into compact shapes, then extend long fishing lines. Portuguese man-o'-wars drift at the surface under wind-driven sails. Members of the dandelion siphonophore family hover near the seabed and use tentacles to tether themselves to rocks. Across roughly 200 described species, evolution has rearranged the same colonial idea into floats, chains, bells, nets, and living chandeliers. Many glow. Blue and green flashes can startle predators or mark a disturbance. Some deep species produce red light, an unusual ability in a world where red wavelengths from the sun never arrive. Transparent tissues make them nearly invisible under natural conditions, while research lights transform them into spectral ribbons against black water. They are not merely curiosities drifting outside the food web. Siphonophores are predators of copepods, krill, fish larvae, small fishes, and other gelatinous animals. They are also prey. Ocean sunfish, sea turtles, nudibranchs, and specialized crustaceans can feed on gelatinous bodies that would be dangerous to less adapted hunters. One crustacean, nicknamed the pram bug, can hollow out gelatinous tissue and use the barrel as a mobile shelter. Their position in the water column also connects the bright surface to the deep ocean. Many midwater animals rise at night to feed and descend during the day, part of the largest daily animal migration on the planet. Species that join this movement carry carbon downward through feeding, waste, and death. Siphonophores participate in a web once dismissed as little more than jelly, but now recognized as a major route for energy through the open ocean. That role is difficult to count because gelatinous animals vanish from traditional samples. Nets shred them, preservatives shrink or dissolve them. Acoustic surveys that easily detect fish may miss transparent bodies with little dense tissue. An ecosystem can contain far more siphonophore life than the collected evidence suggests. The same invisibility that protects a colony from prey can hide its importance from science. This creates a visual trap for us. On camera, the colony looks simple because so much of it is transparent. Up close, it is crowded with repeated anatomy. What resembles a loose thread may carry hundreds or thousands of mouths, tentacles, protective plates, reproductive structures, and swimming bells. The largest creature in the frame can also be the easiest to miss. What comes next is the part that still does not make sense. We have explored only a fraction of the deep water where these colonies live, and even when we find them, we struggle to measure them without changing their shape. The Ningaloo siphonophore was filmed curled into a spiral, not extended in a straight line. Its length was inferred from imagery rather than measured along a captured specimen. The old bootlace worm record came from an animal washed ashore more than a century and a half ago. Jellyfish tentacles stretch, contract, break, and regrow. Every contender turns length into a different biological problem. So the question, what is the longest animal on Earth, has no clean answer until we choose the rules. If we mean the longest reliably documented solitary body, the bootlace worm remains a famous candidate, although its 55-meter record is debated. If we mean the longest massive animal with one integrated body, the blue whale dominates and is unquestionably the heaviest animal known. If we count tentacles, certain jellyfish enter the contest. If we count a permanent, physiologically integrated animal colony descended from one embryo, the Ningaloo apolemia may exceed them all, especially under the higher reconstruction of its full spiral. But ranking them misses the deeper discovery. The siphonophore did not merely add a new name to a record book, it exposed the assumptions hidden inside the record. We picture an animal as a sealed body with a face at one end. We assume one creature means one set of organs. We imagine that cooperation happens between separate individuals after they are born. The siphonophore breaks all three rules. Its body is open and repeated, its organs are modified animal bodies, and its cooperation is built during development. You could call it a colony because it contains zooids. You could call it an organism because those zooids share one origin and cannot survive apart. You could call it a superorganism because selection has shaped the performance of the whole. Every label captures something real and none captures everything. That ambiguity is the payoff. The longest animal on Earth may not be one animal because one animal was never a universal unit. It was a convenient shape we learned from creatures like ourselves. Return to the image from Ningaloo. A pale spiral hangs in darkness while the lights of Sebastian sweep across it. At first, it resembles a single impossible thread. Move closer and the thread becomes a procession of bodies. Move farther away and those bodies become one coordinated hunter again. The answer changes with the distance. That may be why siphonophores feel so alien. They are not violating the rules of life. They are revealing that our rules were local, written by animals with two eyes, one mouth, and a boundary we thought was obvious. Somewhere below the reach of sunlight, another colony may already be longer than the Ningaloo spiral. We may have passed above it in ships, mapped the seafloor beneath it, and never known it was there. Transparent, fragile, and wider than a city block, it would not need to hide. We simply have not learned how to look. Subscribe for the next deep ocean story, because the strangest discoveries are often the ones that change the meaning of ordinary words.